I request all the organizers to please turn off your mics. We are going to start the program within 2 to 3 minutes. So I request all the organizers to just mute the mics. We are getting disturbances.
Hmm?
So we'll start the program within 1 minute. Ladies and gentlemen, good afternoon and welcome back. I'm Dr. Samir Nadaf and I am profusely elated to welcome our speaker of the session, Dr. Sachin Salunke, Manager, Technical Business Development, DKSH India Limited, on behalf of management, principal, and staff of Sant Gajanan Maharaj College of Pharmacy, Mahagaon, to the last session of 4 days online faculty development program. Without taking much time, we'll move to the session. And may I now request Professor Harare sir to introduce our last but certainly not the least speaker for the day, Dr. Sachin S. Salunke, to our esteemed audience. Please, sir.
Thank you, Dr. Nadab, sir. Good afternoon, all of you. Respected speaker, trustee, directors, principal, delegates, teaching faculties, and participants from different colleges and universities. I, Mr. Swapnil Harale, Assistant Professor, Department of Pharmaceutics, would like to welcome you to our national level e-FDP program 2020 on Pharma Industry Insight, Recent Trends, Innovations, and Digitalization, organized by Sant Gajanan Maharaj College of Pharmacy, Mahagaon. It's my great honor and privilege to welcome and introduce our today's expert speaker, Dr. Sachin Salunke. Sir has completed his graduation and post-graduation from Pune University, PhD from Shivaji University, and postgraduate diploma and intellectual property law and Six Sigma Green Belt certification course. Sir is having rich industrial experience in the field of formulation and business development at MQR Pharma Limited Pune, Cipla Limited, Mumbai. He has filed 2 patents and published more than 19 papers and 9 review articles in various internationally reputed journals. Presently, he is working as a manager, technical business development at DKSH India Private Limited for Pan India and Southeast Asian countries including Sri Lanka, Japan, and South Korea. With this brief introduction, I request to our today's expert speaker, Dr. Sachin Salunke sir, to start last session of this 4 days national level e-FDP program. Please, sir.
Yeah, thank you, Arai sir. First of all, I would like to give thanks for the team for providing us me an opportunity to deliver the presentation in such a faculty development program, especially the principal honorable Dr. A.G. Kileda sir, convener Dr. Vishwikar sir, then co-convener Dr. Samir Nadab sir, and chief coordinator and coordinator, that is Galadge and Alman sir, and Kumba sir, and remaining faculty members. So really, I am truly overwhelmed while I received this invitation to give the presentation or the deliberation in this e-faculty development program. And I appreciate that your frenetic and feisty approach to inviting such industrial speakers to deliver their presentation. So without taking much time, I start my presentation. Yeah. So, first of all, I would like to clear one doubt from your mind that during the presentation, you may have heard that I am coming from this DKSH organizations. Most of the members, you have not heard the DKSH. So, actually, it is a Switzerland-based organization. Uh, it has a presence that is a 150 years old organization, that is a Diethel, Keeler, Sieber, Hegner. These are the four individuals. They came together and they started the organization. We are generally providing the excipients to the organizations. We have a well-established presence in South Korea, then US, Europe. Now we are expanding our market expansion services in especially in Asian countries also. So this is a brief presentation, and we have started our operations in India since last 7 to 8 years, and this is where generally the operations of our organization is that providing the excipients to most of the R&Ds in India as well as South Korea, Japan, in Asian countries, giving the technical supports to them, then providing their technical support as well as a to that is from the scratch to that is a commercialization and the finalization of the molecule. So this is our operations. So my topic is that complex injectable drug delivery systems, and it's with reference to its digital and business intelligence. So I assume that I have 1 hour. So in 1 hour, I have segregated my presentation into 3 parts. That is the first 10 to 15 minutes we will cover. That is a generic parts, means there are the different kind of a drug delivery systems in the market. So you know the generic, but here through the point of that is the industrial point of the view, there are the different type of developments which are the going on. So we have to focus on that for the first 10 to 15 minutes. Then after that, for 30 to 35 minutes, we have to give more focus on that is a complex injectable drug delivery systems. So complex injectable means from the scratch of the project. Scratch means from the initial of the development to that is a final commercialization. After that, we will see there is a 3 to 4 proof of concept studies in the injectable drug delivery systems here. And last part, there is a digital commercialization or digital transformations, that whatever these are the happening in the industry. So we will go one by one. So this is main that is a dashboard of our presentation here. So we can see here that is a there are the different kind of things are there here. Through the point of the industrial, we know that that is a IND and NDA and ANDA. So I'm not going to cover that is a much part in case of that is the IND aspects. We will cover that is the NDA and ANDA part for the 10 minutes. Then turnover of whole pharma industry in the market is a 150 billion USD. The number of the plants, if we see here, that is a 1500 plus manufacturing plants with 155 plus market presence that has been situated for the pharma industry. Then there are the different therapeutic segments which are available in the market, like antiretroviral, oncology, anti-diabetic, gastroenterology, and nowadays more focus on that is the antiviral part. There are the different dosage forms are there. Now you know that tablet capsules, but besides of the tablet capsule, there are the much more developed dosage forms, like as in pulsatile drug delivery systems or in complex injectables. There are the microspheres are there, liposomes are there, then long acting injectables are there. So such type of the drug delivery are the dosage forms which are the going on. Then total DMF and ANDA file. ANDA file means abbreviated new drug application. So here in ANDA filing, you if you think that or you can't imagine about Arvindo Pharma in a year, that company suits or the files 40 to 50, that is the US FDA ANDA filings in a per year. So if you see for the entire organizations from the India, so there it goes count that is a more than 1200 or the 1300. Now the history of the successful acquisitions, you know that Biogon, Mylan, these are the going on, went in last 2 years, couple of the years. Then Actavi, Supportex, which are with the Arvindo Pharma. Then regulatory approvals, if we see here facilities, there are the more than 1000 or the 950 plus, which are the in India as well as in abroad. There are the 50 for the Indian organizations like Amnil USA, MSN organizations or the Arvindo Pharma, then Sun Pharma. So they are having a head offices in US also, and R&D also in US situated. And these are the business segment wise: oncology, injectables, biologicals, and the vaccines are also there. Then R&D strength, if you see, that is a 4,000 R&D strengths inclusive of large scale, medium scale, and the small scales, and differentiated portfolio. Nowadays, this picture is going in much boom in the market, like oncology, hormones, depot injections, inhalers, biosimilars, and patches, rather than the tablet and capsules. So you think on this way also. And the PEPFAR program. PEPFAR program means what? This is a presidential emergency plan for antiretroviral products. So this is much going in the market in case of that is South Africa and the rest of the world. So this is also in a boom. So most of the organizations which are the working on the ARV, now you know in a COVID-19 situations, those are the organizations which are the working on the antiviral also, antivirals only. So we go towards next part. So these are the contents that how we can go in this presentation. First part, that is I already cleared out. That is the first part we will cover, that is a generic concept. Then we will see injectable part, and last one, digital intelligence. Now here, that is introduction. You know that generic name and the brand name, we know the difference between the generic and brand name, that is only on the basis of the price. But there is a, by the definition and by consideration of the development, if we see, there are the different differences. Is main is that there is a generic name and the brand name. Generic name obviously, and of course they, it has a low price. Branded heavy, that is a high price. But it is an abbreviated new drug application. Abbreviated means it is a divide of a preclinical study. So this is the meaning of the abbreviated. So that's why is there is a not requirement of a preclinical study for the generic formulation. So that's why we called it as an ANDA, or abbreviated new drug application. We will see in the next part. I'm not covering much part in case of IND, because you know that you have in your syllabus also. So we will cover that is the ANDA aspects. So generally, we will cover in this part, that is a market mapping, or the what is the market value for the ANDA. So there in industry, there are the different type of a drug delivery systems are available. First is the ANDA, that is the abbreviated new drug applications. Second is that complex injectables are there. Complex generics, we can say here. Complex generics means that is a control release formulation in the form of a solid oral. In injectable, that is a complex injectable like liposomes, microspheres, long acting injectables. And third one, more important thing, these are the certain projects which are based on the para filing. So para certifications which are the going on in industry, I think so you may have heard in a regulatory filing lectures also, that is a para 1, para 2, para 3, and para 4. So para 4 certifications are there. So most of the industry is working under this circumference only. So these are the different type of the projects. And fourth type of the projects, are the fourth category which is involved, like as an 505(b)(2) kind of a projects. 505(b)(2) means suppose there is a tablet is available, and if you want to develop capsule dosage form, so it is called as a 505(b)(2). This is in the terms of changing in a dosage form. If we want to see, that is a changing in an indication. So for example, we know that the dexamethasone nowadays going on the treatment, like they have different applications, means antiviral, anti-inflammatory type of the applications. But there is a repurposing of the drug is going on, and we are carrying out the claiming the application as a antiviral. So it is called as a 505(b)(2) kind of the applications. So it comes in the form of a injectable. So now we're developing the dosage form in the complex injectable. So this is like a complex injectable, or the 505(b)(2) we can say here. So if we see here the market, So here market is that 615 USD dollar in 2008, but in 2019 it is near about that is 840 USD. So US generic, if you use that is a cumulative aggregative, that is a growth rate. If you see that is a compound annual growth rate, it is near about 9% to 10%. And in 2021 it is a 19.6 billion dollars. So this is a completely market mapping of this generic market. If you see area wise, so here that is a global generic market. Global generic market here size is that 643 billion dollar, but in generic share it is near about the 55. So growth rate, if we consider for overall market is a 7%, but generic share is a 11%. And these are the key players which are the working across all the countries, and expected growth is a more in case of a generic share. And our Indian pharmaceutical, Indian pharmacies or the pharmaceutical companies are which are mostly based in a generic share. Very few organizations are working in a NDA and ANDA aspect. So we will see in the next slide, but we will see the geographical distributions of this generic market is near about $17 billion for Western Europe, US, Canada $15 billion, and the rest of the world these are the $4 billion. So most of the pharma industries which are the working for the US, Canada, Western Europe, and the rest of the world from India, and some few organizations like Lupin, they are working for the Japanese market also, and the Latin America. So in Latin America, Brazil and other countries which are the comes here. So you know that this is not, I have given that is as a representation, that is what is the difference between generic price and the branded price. So for this only, that is the price aspects I have given. Now we will see in a detail, that is what are the developments which are the going in industrial sector, or what considerations we have to take into consideration for the generic filings. So see here, these are the FDA requirements for the generic drug. Means there is an active ingredient should be same, it should be bioequivalent, dosage form, route of administration, and the indication that should have to be same. Then and then only we can call it as a generic drugs. If there is a tablet and we form the capsules, then we can't call it as a generic drug, because there is a dosage form is going to be changes here. Dosage form and a dosage strength is that should also be same. So then and then only we can say or call it as a generic drugs. Then these are the other parameters also we can say here: identity, strength, safety, purity, efficacy, and quality that should have to be matched. But most important thing to the point of industry is that the bioequivalent. Bioequivalent means that is a bioequivalence criteria should have to be matched there. There are the certain biological study, means whenever there is a tablet is a form in the industry, so that has to be given to that is a clinical populations. So there are the pilot study and the pivotal studies are there. So pilot study means what? There is a pilot clinical trial and the pivotal clinical trial. Pilot it is near about list as per the FDA guideline, you can take near about list number of the patient is near about 12 to 16 patient you can take. But whenever you go to the pivotal trial, at that time you can, there is a no limit for the pivotal trial. But FDA requires this data of a pivotal study also, as well as a pilot study also. Sometimes what happens, I think so, you may have heard the name of the posaconazole drug. It is for the antifungal drug. This drug or this tablet or the formulation is based on that is a HME trial, that is a hot melt extrusion process is there. Now the patent is going to be expired in upcoming 1 or 2 years. So most of the industries like Inventia, Sun Pharma, then Lupin, they are working on the posaconazole tablet. So they are facing the problem that pilot bio study is going to be passed, but pivotal study is going to be failed. So they are facing a challenge. So they again redeveloping the formulations, again carrying out the reverse engineering for that, and doing the filing of the product. So this is the difference. Now, in academic level, we don't give consideration to this IIG database. So whenever you formulate any formulation at your MPharm level or at your research level, so you have to give consideration to the IIG database first. Suppose if you are using your formulation, yes, I appreciate that you are developing certain new excipients from the plants extracts, or you are taking developing new innovations. But whenever you are using these excipients, it's okay. But for whenever you are using these innovative excipients or the innovative formulations with the or the extracts with the other excipients, that time you have to take care that that excipient should have to be in that range. So whenever you go in the USFDA side, there are the certain orange book and the IIG database has been given for the different kind of excipients. Suppose you have to want to use as a TAL or the microcrystalline cellulose. So there are the limits have been given there. So you have to concentrate. Suppose per day there is 86 mg for the MCC, or for cross povidone there is a 20 mg is there. So such like limits that have been given. So you have to take in the tablet not more than that, because it create a complexity or the toxicological problem. So now you go consider this part. So here that is a NDA drugs which are comes under module 3, module 5. It doesn't cover the data that is a module 4. This is a module 4. Module 1, module 2, module 3, module 4 and module 5. So ANDA lies in this range only, module 3 and module 5. It doesn't come under the module 4. Now see here this diagram. Now can you think how now we have seen that in last few slides that is a differentiation in a price of a generic drug and differentiation in a price of the ANDA drugs. Oh, sorry, NDA drugs or the NC molecules. Now see here, but these so if you see here that is a ANDA filing cost, ANDA filing cost, NDA filing cost, and the DMA filing cost. If suppose any organization develops NDA formulation or the NC formulation, at that time they have to file the NDA along with these fees. So this fees is near about the 24 lakhs USD. So 24 lakhs USD. So you can imagine that in rupees it near about 18 crores, 1/8. So here in ANDA, 1 lakh 76, 1 lakh 76 USD. So in crores, it comes near about 1 crore 33,000. So if we take small organization, big organization, or medium scale organization, if they want to file the product as a ANDA, or we can call in a FDA language as a F505J, then we have to submit the fees to the FDA near about this range, is 1 crore 33 lakh rupees. So nowadays, most of the companies which are facing the challenges like mid scale as well as small scale, they are not able to submit the fees of the 1 crore 76 lakhs rupees because there are a lot of insecurities as well as a lot of risk which are involved. So due to that they are facing a losses nowadays due to these situations. And in DM, if we consider for the API also, so it costs near about the $57,000. So it near about 43 lakhs to the 50 lakhs that cost comes. So this is a huge investment these are the going in or going by the organizations in the these US FDA sectors. So that's why everyone taking the precautions that while developing the formulations for the ANDA, they have to go by this route. That is filing the fees as well as providing the data. So these are the challenges we are facing in India. So these are the differences we have already covered. These are main difference is that ANDA doesn't contain this is clinical data, preclinical data. Otherwise, it contains all the data. While IND, you know that IND doesn't contain this microbiology clinical, while it covers preclinical studies only. Now, earlier scenario, you know that earlier before 1984, we have only product patent, but after that, process patent has been evolved in the 1984 due to this Hatch-Waxman Act. So before 1984, there is only one, there is a right to only one innovator that innovator can develop that organization only. But nowadays, after 1984, the laws have been issued and it gives the freedom to other organizations that you can develop other formulations also. So that's why we are that enjoying the rights means many organizations which are the developing their formulations and enjoying their not monopoly but they are enjoying their rights marketing rights in the market. So this is the condition for Hatch-Waxman Act. Now you know that para 1, para 2, para 3, para 4. So I don't think in your syllabus this have or not. But para 1 is that there is a no patent in Orange Book. Para 2 means there is a patent listed, but it is expired. Para 3 means what? There is a patent is there, but patent will expire in upcoming 1 or 2 years or upcoming 5 years. We can say here. And para 4 means that non-para means non-infringement. Means suppose innovator have used that is a HPC means hydroxypropyl cellulose and other organization suppose they use the para 4 means so in this case the para 4s which are the going into the market means innovator if they use the HPC then there if the innovator have used hydroxypropyl cellulose and other organizations suppose they use the polyplasdone or the crosspoidone, then it is not comes under the infringement case. It is a completely non-infringement means it is a completely going to bypass of that use excipient. So these are the benefits of the para 4 strategy. But most of the uh, projects which are the going in industry which are the limited to the para 2 and the para 3 only means patent listed expired and patent are there but will expire in upcoming few days. So these are the these are the concepts which are going on. Now I give there is a brief outline that favipiravir you have seen that from the Glenmark the patent is expiry is a near upcoming 3 to 4 months. So it comes under the para 3. So these are the benefits of developing these projects through the point of that is the industrial point of view. Now these are the top 14 drugs which are the going to off patent in 4 years. So most of the organizations or the pharmaceutical companies which are the working, these are the innovators. But from the India, most of the companies which are the working on these molecules to crack the formula and come into the market. Whenever they come into the market, they can enjoy or they can get this benefit in terms of a dollars. So you can see or you can think on that how much dollars they will get if they crack the formula. So it is a completely rat race. Now you know that that is a para 4 certification. There are the certain court cases maybe the going on for the organization to the organization. So this is I have given the certain strategy that within 45 days, if suppose generic company innovator company files there is a sue or sue means here that is a case against that is a generic player, then the certain cases which are the going on, suppose within after 45 days, if suppose there is a files the case, then there is a no meaning. So in that condition, ANDA applicant granted for the approval. So generic player will get approval because after 45 days that innovator company has not filed any case against the generic player. But if the innovator files the case against this generic player, then there are the two stages may be involved. 30 months stay period is there. 30 months means there is a completely 2 months and half year. 2.5 year. So 2.5 year there is a process is going to be complete going on, that is a toss. So here both the organization have to wait for the 2.5 years. But if this period is expired, then again the decision is depends upon that is the court. If the court says innovator you can go with your formula, then in that conditions that generic player they have to be stopped in this condition. But if the court says that that generic player you can go with your formula, so in that condition first 180 days means first 6 months is given to that is an innovator, and after completion of the 6 months the rights is given to that is a generic player. So this is the condition. If you see 30 months if suppose that is not expired, so in that condition same condition is there, but it depends upon that is a court decision. Suppose that is a court has a given the favor in case of a generic player, then generic player enjoys the right till 180 days. 180 days means you can see or you can think that 180 days means 6 months that company enjoys the right in the market. So there is a no other organizations will market that formulation into the market. So this is a complete scenario of this case. Now we will see that is a case study. Two case studies are there, BMS and the Mylan Pharmaceuticals. Actually, BMS, they have had there is a US patent. So in the 2000, so claims over that is a buspirone as an anxiolytic agent. But before expiry, means 12 hours before expiry, uh, sorry, here there's a BMS, they have the patent, but Mylan, they have formulated their formulations and raised the patent against BMS, and they have filed that is a para 3. This patent is there, but patent will expire. So before the expiry, Mylan has filed another formulation against that is a BMS. But BMS, what has done, before 12 hours, they have filed, before 12 hours of the expiry, they have filed the another patent in the Orange Book. So the whole rights which has been given to here, that is a Mylan and against the BMS. So Mylan own the case here and hold the rights which has been transferred from the BMS to the Mylan. This is the first case study. In second case study, here certain litigation cases has been developed. So Andrex Pharmaceuticals is there, and these are the HMRI. So here both organizations they have settled the litigations. So what the settlement? So HMRI organization they have to give this much amount rupees, that is a 10 million US dollar to that is Andrex for the first 2.5 years, and after that 2.5 years, Andrex Pharmaceuticals they will come into the market with their formulation. So such kind of settlements are also going on if there is a litigation cases happen in the court. So these are the two case studies I have given. Now, to the point of industry point of view, we will go towards that is a bio waivers, bioequivalence, and the bio studies. So you know that bio waivers means what? Bio waivers means there is a no requirement of a bio study carrying out in the formulation. So you know that class 1 and class 3 formulations means class 3 as per the BCS classifications, class 1 and the class 3. Class 1 means high soluble and high permeable drugs, and class 2 is that poor soluble and that is a high permeable. Class 3 is the opposite of that, and class 4 is a poor soluble and the poor permeable. So class 1 and the class 3 drugs, they don't require a bio waivers, they don't require a bioequivalence study, so they can go through this route during the filing, that is a bio waiver. So they can bypass the these bio studies, so they can directly go, they don't require that is a dissolution part and all, but they directly go through these bio waiver studies. Now, in bioequivalence, if we go for the class 2 and class 4 drugs, means there is a poor soluble and there is a high permeable, and poor soluble and the poor permeable. So in that condition, your F2 means dissolution factor should have to be more than 50, and power value it should be lies in this range. So these are US FDA strict guidelines. These are your power value should have to be lies in this range, that is 80 to 125%. Without this, if your range comes below 80, then US FDA can't agree your formulation or they can't agree your dossier. If it goes more than 125, then it goes on the supra side. So in this case also US FDA can't agrees with your formulations. So these are the important points. So bio studies and bioequivalence. Next part, I have not exposed the name of the organizations, but in last 6 months, this acquisition has been placed in Europe organization. It is actually Canada-based organization, but in last 1 to 1.5 year, it goes in the losses. It went into the losses due to this generic competition. But after all, one of our organization from the India, they acquired this organization and they transferred or they expanded their facility in France, Germany, Portugal, UK, Italy, and Romania. So this is one Indian organization like Arvindo. They have generated or they expanded their facility in whole all the countries by acquiring Canada-based organization. So see the importance of our Indian organizations in the market. So here, upcoming or the emerging markets which are the coming into the market, that is a biosimilar market as well as a biopharma sales. Most of the this is a booming market nowadays. So you see the growth of this biopharma to from the 2006 to 2016, and nowadays this is going into the marketing much boom. There is a biosimilars, biologicals, and injectables. So revenue also, if you see the revenue wise and the industry wise, so it is going from the 4 to 8, means it is a double in the year of only 5 years. So there is a 4,800 to 8,400. So it comes into double in the only year of the 5. Now there is a dependency on orals coming down. Solid orals means tablet capsules dependency is going down, but it is going to be more in case of there is the investment in case of a injectable. Oncology means anti-cancer segment, dietary supplements, nutraceuticals. So these areas which are going to be evolved nowadays for the enhancement of their revenues. So if you see, these are the US FDA filings. So one of the organization they have filed, these are the 102 filings, 200 102 US dollars, with filing score is near about there is a 572. These are the some US FDA approvals that received for our Indian organizations in last 1.5 year or 2 years. So these are the some molecules that have been given here, mentioned here, which are some anti-cancers, antivirals are there, and some injections are there, solid orals, and the suspensions are also there. These are the dolutegravir antiviral segment is here. That's the dolutegravir, lamivudine, and tenofovir. Because antiviral drugs they may give the resistance to the patient, so that's why the combination therapy has been evolved. So such kind of approvals are given. So we have covered that is the first part. Now second part is that a complex injectable means what, and we are going from the first scratch to that is the end of the or the commercialization stage. So here, generally, we have handled or we are going to handle in most of the companies this kind of a projects. So I have, or from my team, we have handled these kind of a projects like anti-cancer injections, antifungal liposomal injections, anti-cancer ready-to-use injections. So we will see one by one. These are the how these are the going coming. So first important thing, they are the complex injectable means what, these are the not plain injectable. Complex injectable means it covers liposomes, microspheres, then certain glue-based therapies or long-acting injectables using PLGA. And this is one important thing. These are the lyophilized powder for the solution. So this is the NC-1 project. NC-1 means new chemical entity generally given the rights for the 5 years. So we have to file the product into the market. NC-1 means 5 minus 1 is equal to 4. So within 4 years, we have to file the product into the market. So it has been called as an NC-1. So there is a development is also fast. So now see here, these are the background of the molecule I will tell you. This is intravenous injection, and this is used in case of a cancers. There is a multiple myeloma cancers and a non-Hodgkin. These are the lymphoma, that is a lymph cancers or the lymph node cancers. So it has a high market value into the market. It can be given with the dexamethasone as well as a lenalidomide for the patients. But you see the here patient dosage. Dosage is like that. For the 2 consecutive days, for per each week, you have to give 10-minute infusion. So like this, you have to give the treatment to the patient for the 3 weeks, and last week is a completely rest period, that is a 12-day rest period. So if you take this formulation as an injection, so day 1 2, then 8 9 15 16 for the 3 weeks, and after 3 weeks there is a 12-day rest period is a there, that is a day 17 to 20. Then again next month you have to take a this kind of a treatment. So this is the dosage have been carried out here. Now we will see these are the what are the CMC. CMC means that is a chemistry manufacturing controls are there. Now see here, these are the statement of a problems and known prior art. Actually, in industry, what happens whenever we have to develop the project? So at that time, we have to see that what are the problems associated, or in your research projects also, you are going to see that what are the problems associated with your projects, and what kind of a solutions or the strategies you are devised to give, or the that maybe the plays a remedy role for that projects. So here, statement of the problems and the known prior art is here, that drug is a very slightly soluble in acidic condition. First important thing. Second important thing, that it remains stable at a in a solution at a neutral pH only for the 4 hour at a room temperature. Means for the room temperature that drug is remain in a solution at a 4 hours only. Beyond 4 hours, it can't remain in a solution state. Means it becomes unstable. So to give the stability, there is a requirement of this dosage form to carry out the lyophilization cycle for this. Okay, so this is the main prior art of this API. Next important thing, that there is a long-term storage of a liquid formulation which generally limits the clinical application. So that's why we can say here that the lyophilization is needed, because above that is a 4 hours, it can't remain stable in a solution state. And in next few slides we will see why there is a requirement of a lyophilization, why there is a not requirement of a solutions, why there is a not requirement of a suspension. So we will see in a next part in a chemistry point of the view. And main important thing, that is a very short half life of a drug. So to extend the half life of a drug, we can carry we are developing these liposomal formulations, because lipids which are the giving, that is a slowly release of a drug. So this is the important point. Now what are the hypotheses? What is the hypothesis here? So there is a main hypothesis is that this drug has a proteasome activity, proteasome inhibitor we can say here. So as compared to the healthy tissue, there is a less chances of the to target, that is a healthy cells. Means what happens, actually this molecule has a chemical structure in such a way it inhibits, that is a proteasomes in the body. So there are the less chances of to target the healthy cells, because what happens, there is a main problem in a cancerous drug formulation is that they can target the healthy cells also. So if we develop the liposomal formulation and the target our formulation to only those kind of a receptors, so there is a healthy cells which can be saved here. And main target of these receptors are, there is apolipoprotein E and B, because the particle size comes in the range of 20 to 200 nanometer. So these are the certain strategies. If you see, that is a pictorial here. Whenever we give that is a IV injection by the liposomal formulation, it goes into two ways. First, that is a blood, and another part, that is a lymphatic system in the same way or at the same time. So drug release, whenever it goes by the blood, drug release in a blood capillaries, and here by the lymphatic system. So nanoparticles comes into in the blood this lymphatic system and goes into directly into the liver. So there is a liver causes, there is a metabolism, and then there is a release of a drug into, that is a blood is occurs. So there is a release of the drug into the blood occurs via two ways, that is a blood as well as a directly by that is a lymphatic system. So this is the main mechanism of that is a liposomal formulations. So why we have carried out that is a liposome. Now if we see the particle size of a liposomes, it comes into the range of a 20 to 200 nanometer, like D10, D50, D90, like this is the particle size is there. So these are the nanoparticle liposomes we can say here. And one another approach is that whenever the particle size is less than 200 nanometer, so there is a more chances are there that it can permeate from the vessels also. So this is the main that is hypothesis and the mechanism. Now we will see the main important thing, that is a chemistry. Now whenever there is a formulation development project is comes into the picture, at that time we are only looking towards that is a formulation aspects. But friends, see here, in industries such conditions are not like that. We have to go by thinking its chemistry level also. Means first there we have to see that is a chemistry of that formulation or the chemistry of the API, and then we have to think that what kind of a formulation is suitable for it. So here this structure is divided into three parts, and I have given the physicochemical properties. So log P value, if you see here, that is a 3.77. So you know that if suppose there is a log P value is a more than near 3 or the more than 2.5, so we can call it as a lipophilic drug. So it is a lipophilic drug. pKa value is a 5.14. So if we use the acids, so near about having a pH of that is a 5.526, then it can liberates the drugs in the system if we use the that acids in this formulation near to the pH 5. So it liberates the drugs because there is a common ion effect. So these are the certain chemistry principles which are the involved during this. So these are the two important things are there. Molecular weight is a 719. So it is a small molecule, not like as a small molecule, but it comes into the range of a small molecule and the large molecule. So because it is a tetrapeptide is there. Okay, now we go towards that is a chemical structure. So here there is a morpholino group is there, tetrapeptide and epoxy ketone. So we have to see that what kind of a degradation pathways which are associated with this chemical structure and how we inhibit this degradation by considering the formulation approaches. So we will go one by one. So this is a chemistry review. We have already just I have given that is a outline that these are the four chemical three rings are there, morpholino structure. Then there is a tetrapeptide and epoxy ring. Okay, so these are the involved here. Then we will see in next slide and just information point of the view I have given that is a this administration period also, because why I mentioning on this slide, because this information have to submit to the US FDA while developing the product. So now you see that is a chemistry review we can say here, or there is a chemistry manufacturing control also we can call it as. Now there is a peptide hydrolysis. Now these are the degradation pathways. So why we are going to carry out or why we are going to understand this degradation pathway? Because if we see the degradation or the understand the degradation, then we then that degradation pathway will give or the denote us that different kind of approaches to inhibit those kind of a degradation. Suppose, for example, here NHCO group is going to be hydrolysis in presence of the acid and the water. Suppose you have prepared suspension or the tablet, and in that suspension there is some amount of the acid or the water is there. So due to acid and the water, what it affects? So it gives the effects that it causes the hydrolysis and it forms this kind of a compound. So that is a morpholino homophenylalanine leucine group is a form and homophenylalanine. So these are the two groups which are the breaking. So this group is going to be break this form that is a homophenylalanine, and this group is going to be break it forms that is a leucine compound. So it doesn't give the actual impact of this ring. So what? So this is the culprit that acid or the acid hydrolysis or the presence of the acid or the any water molecule in that formulation. So it liberates the hydrolysis. So we have to prevent that. Next important thing, you know that is a reaction mechanism. So this water molecule attacks on this carbon. Here nucleophilic substitution is occur. Then lone pair of the electron goes here and it carries an unstable part. So at last at finally what we get here, that is a carboxylic acid and the amines we will get. So we have to prevent this. So to prevent this, we have to use to divide of such chemicals in such a way that it divide of the acids or the water. So this one clue we got that we have to use the chemical that is a divide of the acids or the water. So next we will move to a next part, that is epoxide hydrolysis. Now epoxide hydrolysis, last ring that is the epoxide ring is there. So whenever there is a nucleophilic attacks on this, so here same way is a form, that is a nucleophilic substitution is occurs and there is a diol ring is a form. So see here that is a diol ring. So now in next slide we will get idea why there is a requirement of a lyophilization. Now, see here, to minimize the pH or to minimize the acid or the water, there is a requirement of these strategies. Whenever we take a solvent, there is a dielectric constant that should have to be less. So there is a less activity. So there is a less interaction chances which is occur in that formulation. So we are preparing ethanol, methanol rather than the water, rather than the dimethyl formamide, because water has a high dielectric constant as compared to ethanol and the methanol. We can use this also, benzyl alcohol also, because it has a less dielectric constant. Dimethyl formamide also we can use because it has a less dielectric constant. So use of a dielectric constant should have to be less. Now esterase inhibitor also we can use in our formulation, that is a EDTA, peroxone and the sodium fluoride also. And pH. pH is the most important thing because we have seen the pK of this drug is near about the 5 to 5.14. So we have to use the chemical in such a way that it liberates the drug at the near to the pH of the 5 to 6. So we have to select the pH in that range. Okay, and temperature should have to be cold because it divides, it removes the water and it frees the environmental conditions or the environmental molecules in the free state. So there is a no liberation of the water molecule in the formulation. So these are the strategies to prevent the hydrolysis. Now I'll give explanation here why there is a requirement of a lyophilization. Now we see here the different kind of impurities are there in this formulation. We have studied this. The diol impurities are there, N oxide impurities are there, epoxyamine impurities, then epoxyleucine, alkene impurities. So much kind of impurities are there, and as per ICH Q3, Q2, and Q6. So there is analytical method validation and there is a Q6. These are the some related to that is the impurity. So these should have to be in a limit. That is a threshold potentials. It comes below the threshold potentials. So if we see that the diol impurity, so it should have to be less than that is a 10 ppm as per the US FDA and the monograph, USP monograph. Now we see here why there is a requirement of a lyophilization, because whenever there is a diol impurity goes more than the 10 ppm in the formulation, at that time what it happens, there is a drug remains unstable, because increases in diol impurity amount when the drug in solution state, so there is a instability is a form, and so that's why there is a requirement of a lyophilization. Because if we suppose develop the solution or the suspension, so if we develop that kind of a formulation, then there are definitely 100% sure that there is a diol impurity which are going to be increases. So if it diol impurity generates, then it becomes unstable to the formulation. So that's why there is a requirement of a lyophilization. So this is a need of a lyophilization. Next important thing that alkene impurity form at a highly acidic pH conditions. Highly acidic means the pH should have to be less than near to the range 3 to 4. Whenever we use such kind of a chemicals, so alkene impurities generates. So we have to avoid that. So we have to use that is a weak acids for the pH adjustment. So we will see the next part. That is the API solvent compatibility study. These are the pre-formulation study we have carried out. So in this solvent we have used here that is a chloroform API, all the impurities which are under limit. So we can take the chloroform for our trial. So there is no impurity detected and compatible solvent. If we take second part, ethanol. So in ethanol, there is a certain kind of impurities which are developed, but we can give the reasons for the of justification that it due to that is a degradation mechanism is oxidation, and in oxide kind of impurities may be formed, though it has a less dielectric constant, but it can't be used because it gives the impurity by the due to this oxidation degradation pathway. Next part is that parentally acceptable. We have seen that there is a lipid should have to be pH near to that is a not pH, but there is alpha hydroxy acids, which are the comes in this lactic acid and the tartaric acid. Why these are the requirement? Because it aids or it helps for the drug to remain in a solubilized state. Because before lyophilization, that formulation should have to be solution. So to become solution, lactic acid and the tartaric acid, which are the helpful because these are the containing alpha hydroxy acids and they maintain the drug in the solution form. Other acids they don't maintain, they can't maintain the solution drug in the solution form. So we have to avoid this, but we have to take this lactic acid and tartaric acid. Now see here, this is the procedure, main procedure. API is used, DSPG sodium. DSPG sodium means what? This is a lipids is there, the diastereoyl phosphatidylglycerol. It is a negatively charged. HSPC hydroxy, there is a hydrogenated soya phosphatidylcholine, cholesterol, alpha tocopherol, sucrose, and these are the chemicals, pH adjustifying agents. Now the beauty of this formulation is that we have to decrease the pH at this range, because at a less pH near about 1 to 3 pH, there is a less degree. Means what happens at a less pH, there is a ionization is happens here. DSPG sodium, DSPG minus and the sodium plus is happens, and a drug contains negative charge. So drug contains negative, this positive, drug contains positive charge. So drug positive charge and DSPG sodium minus negative charge. So it forms that is a electrostatic interaction and forms their bonding at a less pH. But after that, we have to increase the pH. Why there is an increment of the pH is occurs? Because whenever we increase the pH, there are the less chances of that is a liberation of a drug from the lipid cavity. So that's why we are going to carry out that is increase the pH of the surrounding. So this is the completely process. So we have to go to a rotary evaporator, or in case of a on industrial scale, there are the large rotary evaporators are available. Then formation of a lipid films. Then there is a homogenization and lyophilization. So these are the certain manufacturing flow rates that has been mentioned here. Now US FDA, what they demands here. Now we have formed that is a liposome formulation. We have carried out particle size. We have developed lyophilization of that. But US FDA needs this study. That is, study is required human plasma at a 37 degrees Celsius for 24 hours. Why there is a requirement? Because whenever that formulation goes into our bloodstream, their temperature is near to that is a body temperature. So that's why they required. They want to check that in human plasma, are there any degradation happens of this formulation for 24 hours? If the degradation happens, then they can't agree or they can't take this formulation ahead. So we have to that's why they need this human plasma study. Then pH 5.5, 6.5, 7.5. So can you tell me why there is a requirement of 5.5, 6.5 and the 7.5 three-tier system required for the US FDA? So anybody can you tell or why we are going to carry out this kind of studies here? So here that is a 5.5 means acidic pH, I think so. And most of the cancerous drugs which are the acidic. So this 6.5 is related to that is the outer covering or the outside or the outer membrane of the acidic that is the cancerous cells which are the lies in this region, and this is a completely blood pH. Okay, so they need the study in all pH. Is there any degradation or is there any activity happens or not? Next important thing that is a temperature 43, 47, 52, 57, and pH 6.5 upper. pH 6.5 is near to the cancerous cells environment, where to see that there is a requirement or where to see that there is degradation happens or not. So this study, four study required by the US FDA, and ultrasound required for the 2 hours. So can you, anybody from you tell me why there is a requirement of a ultrasound study for the injectable formulation? Because this is an in vitro study, and why the US FDA demands this data? So anyone of
Sir,
you, can you tell?
sir, actually participants would be muted.
Okay,
So
okay,
at
no
the
problem.
end,
Okay, no problem. So here, main important thing is that during the transportations, what happens? There are the certain leakage or there are the certain tendency of there is a floating of the particles happens. There is a unsaturation or the saturation or the precipitation that may be happen in the formulation. So to check such kind of happenings which occurs during the transportation, so they can carry out the ultrasound study. And this is a in vitro bioequivalence approach. So these are the mostly carried out in inhalation formulations. Now we will see that is a QBD aspects. QBD means that is a quality by design aspects. There are the different kind of things are there. So that is a quality target product profile is there, risk mitigation is there, and a risk identification. So these are the three stages of that is a QBDs are there, and QBD comes under the part of that is a digitalization also. That is a whatever the our motto of this FDP is there. That is a digitalization. So it comes under this part. So stage one, stage two, and stage three. Stage one is the quality target product profile. Means suppose for example, I'm just give you that is a simple example. That is a tablet is there. So in tablet there are the 4 to 5 different end results are there. Like for example, tablet weight, friability, disintegration, dissolution. So we can call it as a QTPP. That is a quality target product profile. So this is a one quadrant. Second quadrant is that risk identification. Risk identification, now in this formulation we have identified the risk, that is acids presence, water presence, or the pH. Suppose if there is a less, then it can make a harmful to the substances. Or suppose there is a dielectric constant of a solvent is go more than the 50, then it can create a impurity generation. So these are the risk identification and the risk mitigation. So risk mitigation we have also seen in this section, that is the how we have to cover the risk mitigation, that using of a solvent dielectric constant near to that is a less, that is a less than that is a 50, or that is a divide of the chemicals which is a less than divide of the chemicals which is a less than of that is the acidic pH. The pH should near to the 5 to the 6, or the carrying out of that is a lyophilization process. So these are the risk mitigation approaches. So this is the QBD design that is also comes under this or we can say here that is a digital platform, the digitalization or the digital innovations which are going in the industry. And this nowadays this data not needed by the US FDA, but most of the organizations they are preparing this data for as a support purpose. Now we will see for this liposomes, there are the different kind of a quality target product profile. Particle size is there, zeta potentials are there. Now DSPG, there is a negatively charged in the presence of acidic pH. So this is a risk is there. So we have to avoid that risk. And the process conditions. Process conditions means that is a rate of that is a revolutions per minute we can say here. Temperatures. So this make an impact on that is a formation of a lipid films during the formulation. So these factors we have to take into considerations. Then whole time study is also carrying out, that is a whole time for the 4 hour, 8 hour, and 24 hour for the 60 degree Celsius temperature, because our formulation most of the going most of the times what happens, there is the target country, for example South Africa, there is temperature is the most on that is a higher side. So at that time we have to see that it gives that is a certain impact to, or it remains stable at that temperature or not. Not there is temperature is 60 degrees Celsius, but sure there is a temperature is more than 45 degrees Celsius. So in that condition we have to see the effect. Then photostability study, and in injections there is a one more important thing, that is the tube and the filter compatibility. There are the different kind of tubes are there. There is Advanta POs are there, silicon tubes are there. So we have to these are the coming from, that is the Merck and other organizations. So we have to see that the compatibility of our formulations with these filters and the tube. In filter compatibility is also whatever the filters. Now you have seen the filters for the analysis purposes. There are the zero point 0.2 micron or the 2 micron filters are there. So that formulation is that filter is a merge in that formulation, and for the 24 hour, and after 24 hour that formulation is removed from that filter, or the filter is removed from that formulation, and that formulation is taken for the analysis, that are there any leaching or are there any incompatibility has been happened with the filter with that formulation. Because during the injection you know that that plunger volume or plungers, these are the coming into contact with the solution. So that's why we have to carry out this study. Then pH adjusting agents quantity optimization, and the sequence of the addition, stirring time and the speed. So I have given one example, that is a posaconazole. It is not giving, that is a failed study. So most of the companies also which are the working in this sector, that is a sequence of the addition, stirring time and the speed. So injectables also this care has to be taken into the consideration. Now solvent evaporation temperature 50 degrees Celsius, 60 degrees Celsius, and amount of a glass beads, that is also optimized. So here that is a weight is a 157 gram and the 100 gram with evolution. There are the certain reasons, because suppose at this load it gives, that is a uniformity lamellarity for that film. But if we give that is a low load, so at that time it can't give, that is a lamellarity. So these are the reasons, that's why we are carrying out the QBD. Optimization of the HPS, high pressure homogenization. So we are going to carry out the optimization results, means impact of 500 bar and the 200 bar on the particle size. If we give that is a higher bar, then it gives less in particle size, but it should have to be stable. If you give that is a low bar, then it gives that is a more particle size. Then same part, that is a filtration study. Filtration study means we are carrying out, that is a maximum filtered volume during the filtration, because in at a large scale in industry there are the more filtration capacities near about 6 liter to 10 liters are there. So in that condition we have to check that it gives the filtration filter or the filter that volume at for that rate or not. And lyophilization in this condition there are the three stages are there. Freezing is there, primary drying and the secondary drying is there. Freezing cycle is 34 hour, primary drying 156 hour and secondary 7 hours. So it is actually expensive process, and it goes near about 4 to 5 days. For the it requires the time to dry that formulation near about 4 to 5 days. So these are the process which are the comes under this. But it gives, that is a good water content assay, entrapment efficiency, particle size distribution, reconstitution time. So I think so if you have work in your practicals, or you whenever you are giving the practicals to your students for the liposomes and all, so you can get idea about the particle size, entrapment efficiency, reconstitution time. So these parameters which are related to that part. Now, what are the problems which are associated with the lyophilization? Now, most of the times what happens, there is a while breaking is occurs. Why this is occurs? Because this during the drying or the freezing, what happens, they it the particles in this formulation, they are the rearranges during the drying, and it gives forces on the wall of the container. So due to these forces on the wall, there is a sudden breaking of this vial is occurs. So we have to take care in this primary drying also. Then next part is that if suppose if we maintain the higher supercooling rate, so in that condition what happens, if the cooling rate is higher or the supers, so at that time there is a longer time is required for the drying. Because if suppose there is a cooling rate is a maximum, at that time there are the maximum very fine crystals are the form, and it takes times more time to dry or the evaporation of the water from that cavity. So it gives, that is a maximum time for the, or the longer time required for the primary drying whenever we keep that is a super cooling rate or the higher rate of the cooling. So in that condition, we have to take care of that. That is a control nucleations. Means we have to adjust primary, that is a drying rate, or we can say here chamber pressure, freezing ramp rate, and annealing time. These are the certain process parameters. Whenever you see the freeze drying apparatus, at that time you understand that how we can monitor our formulations process with respect to getting the results as on the accurate level. So these are the parts. Now I'll give, that is a certain we will, so this is a one part, that is how we are going to develop our formulations. from the scratch level to that is the development. Now, whenever we get the better results in terms of the particle size, entrapment efficiency, impurities. At that time, we transfer this technology to the market or the that company. Now these are the proof of the concepts or the some injectable formulations. So I have taken that is a pegylated liposomes. So pegylated, why there is a requirement? Because pegylated means PEGs are there. So PEG, it gives that is a hydrophilic environment to that is a formulations. And whenever it goes into the body, at that time body's macrophage cells as well as other defense mechanism, they can't sense this formulation as a foreign. So they take it as their own formulations, means they can't consider it as a foreign bodies. So they can't recognize the signals from the defense mechanism. So due to failing of this signaling of these mechanisms, this formulation directly goes into the bloodstream and it remains circulated for the longer period of a time. So these are the certain case studies has been given. Cytotoxic drug and antibody is given here, and how these are the attached to these molecules. So this is a case study because these are the PEG linkers are here. So these are the polyethylene glycols. So PEG, these are the links these cytotoxic molecules to the antibody, and it gives accurate release or the actual release of the drug into the body. So these are the certain case studies and the molecules that have been given here. So next important thing, that is this is a second case study is there. Actually, you know that that is a ibrutinib. It is a oral capsule is there. Capsule dose is if you may get surprised that this capsule dose is a 560 mg, but you have to take this capsule 4 times a day in a day. So you are taking anti-cancer drug near about 2 grams per day. So it is a very heavy. So actually, if we see the actual formulation's bioavailability is near about 5.8%. So whenever 560 mg at the body gives, so the 5.8% is near about to 32 mg. So if we formulate, or we are thinking that if we formulate this formulation in such a way that instead of the formulating tablets, if we develop the injectable formulation which gives 100% bioavailability, so why there is a requirement of that is a large dose? So we have taken the consideration that is a 32 mg dose, and it will use the depot type of the injection that once a week is where to give the injection on today. So next injection will come on the next week, next Monday. So like that, this has been developed. And if you see that is the cost of this formulation is near about, this is a capsule cost and this is a local injectable depot formulation cost. This is going near about, that is a 1/4 or 1/3. So we are developing this kind of also formulations, and they given the support to the most of the organizations. Third concept is that so this is the pictorial presentation, just we have seen in case of a liposomal formulations. It goes from the but here we have taken the formulation as a nano suspension instead of other liposomal formulations. Now case study third, that is a ART therapy, that is antiretroviral therapy. Just same part, this is a oral formulation and we have tried to formulate into the injectable, because what happens whenever the AIDS patients or the HIV patients, they are taking the formulation for a longer period of a time, so their oral cavity is going to be reduced, so they can't tolerate, can't take more than the 50 tablet. And there maybe the chances of there is a fungal infections, candida species, which are the formation on the esophageal level. So to avoid that, if we develop the injections in the terms of the microemulsions or in the terms of the microspheres, so it can be helpful. So we have developed the strategy and the working on that, and some our researchers went to on the side of that is a pilot scale also. So this is a proof of case, proof of concept for the case studies we can say here. Now, here proof of concept studies fourth, that is the using of a non-aqueous solvents. Now we have seen that the aqueous solvents may create a problem of that is impurity, or may create a problem in generation of the unstable of the formulation. So instead of the aqueous solvent, if we take non-aqueous solvent, what will be the effect? So there is no effect. So in that case, we can take non-aqueous solvents, and there is no need of a lyophilization also. So it hinders or the avoids the cost of that is lyophilization as well as a impurity problem also. So these two problems which will be removed at the same time. So we have taken that is dimethyl acetamide as a non-aqueous solvent to remove this one. Now we will see last part, that is a digital innovation and business intelligence. So here there are the certain concepts, I think. So digital innovation and business intelligence, it relate to the from your academic to extension into the industrial. So academic, you have seen that is a GraphPad Prism, I think so you have used. Static software also you have used. You have developed 2 factorial design, 3 factorial designs. There are certain different kinds of you have utilized. But in industry, it goes more on that is the extension side. So we will cover those points. Now why I have introduced that is a 4.0, because now industry working on that is a 4 point someone study is there. Now this concept has been developed by Mr. Achyut Godbole and Sudhir Deshmukh. So they have developed this 4 point concept. They both they are working on IBM, Patni, L&T, and making the efforts to carry out. And Bhushan Kelkar, Dr. Bhushan Kelkar also they have developed this technology. So and they are working on that is a utilization of the artificial intelligence in the pharma sector. So see here, that is I cover the part into three parts. First part we are covering that is a digital marketing, then actual utilization of a digital platform in case of a formulation development or the R&D, and how we have extended this academic excellence into the industrial. So these are the digital marketing concepts are there. So for example, TAG linkers to the antibody engineering. So we are working on this actually. So we have already covered that is antibody is there, cytotoxic drug is there, linkage is there, and there we are one of the organization is the NOA from the Japan. They have synthesized, actually synthesized, these are the chemical synthesis process. They have synthesized these kind of a molecules, these excipients for the attaching with the APIs. And it depends upon different kind of a molecules. NH2 molecule is there, NH molecule is there, CONH linkage is there, malimide linkage is there. So they're working on these kind of a molecules to attach the APIs with this and the liberation of the drugs into the body. So this is the one important thing we have marketed through our digital platform. We call it in our organization as a low cost system. So we got a better results from the that is manufacturer. They are more interested in it. So from India, there are the Nectar Therapeutics, Mylan, Biocon, Sun Pharma, Cipla, and Intas. So they are working on this concept, and Sanofi Aventis also. So they are working on antibody drug conjugates, and this is a completely antibody engineering. So they have taken the efforts by the digital engineering only. Next important thing, that is application driven by the enzyme engineering. So enzyme engineering is also there. So this is the antibody drug and antibody drug conjugate. There is a cytotoxic drug, but here there is an actual drug is there. Now here stages which are the liberated from the screening, fermentation, downstream processing, and the final formulation. So these are the different stages are there. Beta lactamase, there are the different enzymes are there. Beta lactamase one of the, is that one is there. Phospholipase, peroxidase. So these enzymes which are the involved during this synthesis. And we have also carrying out this is from the Euclidis Bioscience Austria. We are working with these organizations. We are taking the excipient from these organizations and supporting to the Indian R&Ds. Next part is the enzymatic drug conjugation. This is just a theoretical part that have been given. Uh, that is the antibody fragment segment we can see here. Then covalent conjugations, antibody drug conjugations, and ELISA. So nowadays, COVID treatment is also working on the same line. Antibody drug conjugates, ELISA treatment, then plasma treatment, and next important thing, there is the Moderna Therapeutics from the USA, then Biocon, then Cithorus, then Coriolis Pharma. These are all from the US. They are working on this, and Lonza Biopharmaceutics. So all these organizations, these are the working on biologicals. So next important thing, they are the bioresorbable polymers for the parenteral, means injectable but long-acting injectable. So here PLGA and the PLAs which are the involved actually in this case giving the long-acting injectable. Now see here, this is a digital innovation in actually in formulation development. So if we see here, lactic acid and the glycolic acid, these are the two main ratios are there. Whenever you increase the glycolic acid ratio, so see here in this table, whenever you increase the ratio of the decrease the ratio of the glycolic acid and increase the ratio of the this lactic acid, at that time your liberation of that polymer with the drug into the body is going to be enhances. So it enhances from the 0 days till 90 days. 90 days means till 3 months it going to be deliver the drugs into the body by variation in the concentration of these molar ratios. That is the lactic acid and the glycolic acid. And these are the completely program monitor and digitally monitored to give the drug delivery. Means it is a completely based, this synthesis is completely based on that is a digital monitoring. Next part is that I have given certain I have given certain I have given one example that this is a tablet, this is the injectable formulation, and now this is a tablet formulations. Now in tablet formulation, what happens? There is a tablet information, drug information, and the dissolution data. So these are the three different parameters. And what kind of a study or what kind of a data is required for the digitally monitoring? So here from the drug information, there is a solubility, glass transition temperature, log P, pK value, and percent loading. This is required to input the data to the computer system. Now, what kind of a tablet information is required? Weight, geometry, hydrophilicity, stability, dissolution. You know, that is a fasted steady state, that is a steady state intestinal fluid, we can so call it as, or the fasted simulated state intestinal fluid, we can say here. So this study of a dissolution in this FASIP and FASIP is required for the 24 hours also required. So it gives the pattern of a drug release like this. So if you see that is a green dots, it is the actual release, and this is the red dot, it is a desired release. Why this is a desired release is carried out? Because there are the multiple data that has been fed to the computer. There are more than there is a 10,000 trials are there. So depending on those trials, the computer generates such kind of a predicted release, and we have carried out this actual release. Now we should see that what kind of a exact matching of this actual and that is a desired release. So it gives exact matching near about 95% confidence interval, we can say here, if we apply the statistics here. So it gives that is a exact matching of the desired release and the actual release. So instead of the carrying out the different kind of a trials or the number of a trials, it saves our time from 3 months to only 1 month, or the 1 month to the only 4 days. So there is only near about 26 days that has been saved here to develop the formulation. If we have a data of tablet information, drug information, and the required dissolution study. So these are the new innovations which are the going in a digital marketing. And this is I have given there is the molecular modulations. So maybe quantitative structure property, like as in a chemistry, you have seen that quantitative structure activity relationship, but it is a related like as in a quantitative structure property relationship. But it is a much beyond of that, because it taking the information of all these physicochemical properties of a drug. They have the output data and they are the correlating with this input output with desired and the actual release. So these are the actual innovations which are the going in industry with respect to this digitalization. Okay, now we came to the last part. It is out of scope, means from my side I have covered all the parts related to the digitalization, means what and digitalization, then complex injectable, and the generic formulation. So how it is a relevance to the each other. So we have covered. In digitalization, one more thing I would like to cover that we have covered that how the digital marketing it goes beyond, or how the customer gives the response to you. So generally, we are from our organizations, we are doing the digital marketing, and customer giving the reply whenever they click our digital marketing part. So you have seen the word like as in a Google ads or the LinkedIn campaign. So whenever you click on that link, so at that time we get the report that this person has this kind of inquiry, he is very interested. Then we are again calling, we get the email ID of that person. So we calling to that person and getting all the fetching the information that which what kind of information he needs, he or she needs. So see, such kind of a digital marketing are going on. We have seen this slide that is with relevance to that is a formulation development. And last part I have forgot to tell you that how the academic digital that extended to the industrial part. Now in academic, you have seen that you have used that is a statis graph, this kind of a softwares for the your statistical significance. Now if we go to the industrial sector, there are the different kind of strategies. For example, I'll tell you, operational excellence department is there. Operational excellence means these statistical techniques extend towards that is operational excellence. Operational excellence, it covers the statistical technique like the Minitab is there, Kaizen is there, statistical different intervals are there, 90%, 95% confidence intervals are there, Pareto charts are there, then three-dimensional charts are there, then student t-tests are there. So they are involving these parts comes under the operational excellence. Second part, we can say here Six Sigma green belt, Six Sigma black belt. Six Sigma green belt means what? It contains statistical plus green belt. Green belt means what? DMA DV or the DMA IC, we can say here. So there is a define, measure, analysis, improve, and control. So these are the 5 process parameters which are the comes under the Six Sigma green belt. Six Sigma black belt, what it does that is a DMA DV. DMA means design, measure, analyze, define, and verify. So the other 5 elements comes under the DMA Devi. So operational excellence, green Six Sigma green belt, Six Sigma black belt, and fourth important consideration here, that is a change management, program management, or uh, we can say here that is a portfolio management. Portfolio management means their main duty in the portfolio management is that they have to look on each and every sector. Like as in, they have eye on, that is a formulation development department. They have eye on that is a research and development department, QA, QC, regulatory. So they have to take a follow from the each and every department on a weekly basis, and they have to transfer or they give the expedite that process of that project to that is a finalization state. So these are the different kind of the departments. Now we can say here this is out of scope, so I have covered from my side. There are the different type of there is a digital approaches. Now there, this is the last slide. This is out of scope from our part, but this is definitely useful for you and your students also. Nowadays what happens there, if you see here, there are, these are the only 3 avenues which are we are giving to the students. Because I am a very proud, I am feeling very proudful, and it's my privilege that I am working in a pharmacy profession. Because nowadays you understood that in COVID situations also it is a completely depend upon that is a research. Yes, of course, that is a doctors, engineers, they are helping us, but it is a complete, our base is that that is a doing of a research on a molecular level, or a that is a doing a research in a pharmaceutical or the biotechnological sectors. No one is able to develop the vaccines. So through the point of a pharmacy profession or the enhancement of our career avenues, or the to guide our students, I'll tell you these are the different avenues which are the available into the market other than this. So I think so the days has been gone now to think on these sectors only, that is a production, QA, QC. Now we have to deliver or the motivate our students to come into this sector. They are awaiting, these are the awaiting career avenues, and they are literally, our students, they don't know that what kind of a opportunities are there in the market. Suppose for example life cycle, life product cycle services are there. Now other than the pharmaceuticals, there is I think so you may have heard the name of that is a Deloitte, Indegene, then another organization, Novartis, you have heard, of course. But Deloitte and Engine, these are the certain consultancy services. They are basically or the completely working on the life product cycle services, conjunction with regulatory and the pharmacovigilance. Business intelligence, you know that business intelligence, business analysis position is there. These such kind of positions are available in a GSK, Eli Lilly. So business intelligence are also there. Techno-commercial, that whatever I am doing here, there are some companies they need that is a PhD graduates or and having the background of that is a R&D. So they work on that is a technical matters as well as a commercial things, market mapping also. So it will be also helpful. Complex generics R&D, generic R&D are the working, but complex generic very means most of the organizations which are the working on complex generics also. So we have to give concentration to here also. And then portfolio strategy, I have already delivered, or I have already explained. In the case of that is a digitalization, there are the portfolio and product strategy, or the program management is there, Six Sigma is there. So these are concepts which are also in the market, and they are awaiting for us. And last one is that is the health economic outcome research. Health economic outcome research, this team is completely now means in US this is a completely in boom, but nowadays in India just it has been started in most of the organization, because it combines the marketing information, taking the information from the market and giving that data to their R&D team. For example, nowadays everyone is working on the antiviral molecule, but nobody is working on that is the antibody drug conjugate. Means working, but there are the very less strategies are there, antibody drug conjugates or certain dendrimer technologies. So such kind of a team, they are the focusing on the need of the patient, and the transferring the information from market to their R&D team. So the R&D team, they can work on these type of the projects, and they develop the projects from their uh, the main their R&D stream, we can say here. So these are the avenues which are available, and I hope definitely this information may be useful to you, and you can transfer this information to your students. So I have completed from my side. So if you have any questions, then you can ask.
Thank you, sir. It is always a pleasure to listen to you, and thank you for giving your valuable insights on innovations in complex injectable drug delivery systems with reference to digital and business intelligence. Now we'll start with the question answer session. I request Harari sir to proceed further with question answer session and propose vote of thanks. Please, sir.
Thank you, Dr. Nadab sir. Now we will move forward to the question answer session. We will pick some important questions which are relevant to the topic from YouTube chat box and also from interested candidates who is in Zoom live session. So if anybody wanted to ask the question, please raise the hand so we can
Sir,
transfer the question to the expertise.
Sujit sir has raised. Sujit Desai.
Okay. So Sujit sir, I am going to unmute you. Please ask the question. Sujit sir,
Hello.
please
Hello.
ask your question.
Okay. Good evening, everyone. Sachin sir, am I audible?
Yes, yes.
Okay. First
Yeah,
of
you
all,
can do.
a very nice session delivered as usual. My I asked two questions. First one is
Yeah.
which nano system is more stable, that either steric stabilized or electrostatic stabilized?
Actually, if we compare the steric stabilization with the electrostatic, uh, we can't say like that. But steric hindrance gives that is a more stability because there is a less chances of that is a instability or the less movement of the chemical bonds or the chemical structure. So steric stabilization definitely gives that is a more stability. But if you are focusing on the electrostatic, then you have to cover or you have to focus on the other components also. Means like as in a solvents or other excipients, so or the certain pH adjusters, so they can't elevate your system pH of your systems on the more side and becomes unstable. But in this case, that is a steric stabilization is a more important. It gives that is a more stability against your electrostatic.
Okay, okay. Thank you, sir.
Yeah, yeah.
Whatever you had covered the last part of your presentation, that is the
Hmm.
digitalization in pharmaceutical industry, that is the
Yes, yes.
4.0.
Yeah.
Really, that is the in academia is and also in industry 4.0 is now need of ours in this pandemic
Yes.
situation. We had
Right.
also learned so many things. That is the ICT teaching learning process, new ICT tools, whatever the OBE and number of facts. So whatever I have, we had also in my research work prepared the software, that is the time
Hmm.
required to get a 90% dissolution, that is the QSPR,
Yes,
quantity
yes, yes.
structure property relationship.
Yeah,
So
yeah, you
whatever
have. Yeah, yeah, you have done a lot of research on that QSPR. So
so
definitely,
what
because nowadays, if you go to the one Ashland site, Ashland USA, so he is mainly working on the excipient development. So they
yeah,
are
yeah,
working on this, that is a program delivery and working with the QSPR. But they are not
yeah,
calling it as a QSPR, but they are calling it as a CR wizard, that is a control release wizard.
yes, 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 yes.
Yes,
Thank you very much, sir.
yes, yes.
Hello. Anybody want to ask the question? Please raise your hands. Yes, anybody?
I request Harari sir to proceed with vote of thanks.
Okay, Nadab sir. On behalf of everyone here, I would like to thank you for more interesting presentation. I would like to express our appreciation of your most interested talk on innovations in complex injectables, drug delivery system with reference to digital and business intelligence. Sir, you have been given a very informative knowledge, and this will definitely helpful in future to our participants and delegates who participated from various colleges and universities. I am also thankful to all delegates and participants who were present for this entire knowledgeable session. Sir, thank you for accepting our invitation as an expert speaker for this national level e-FDP and share your great knowledge and views through online mode. I would like to thank the organizing committee, teachers and staff members for their enormous cooperation. Once again, I am thankful to you, sir. Thank you. Thank you very much. Now I am handed over to Dr. Nadab, sir.
Thank you, sir. Now it's time to proceed with valedictory ceremony. And I also request Dr. Sachin Salunke, sir, to be with us for this valedictory ceremony. Dear participants, Newton Baker has rightly said that the man who graduates today and stops learning from tomorrow is uneducated the day after. Keeping this in mind, we have been listening to eminent speakers deliberating in the areas of drug delivery systems and digitalization. All these valuable insights have gave us a new perspective that one should adopt while discussing these issues. Now I call our beloved principal, Dr. Suresh Ji Killedar, sir, to address the participants. Please, sir.
Okay, thank you, Samir. Am I audible?
Yes, sir. You are audible.
Okay. Thank you, Samir, sir. And first of all, let me congratulate our today's speaker, Dr. Sachin Salunke, sir, who has enlightened so many things in the development of injectable dosage form. So let me congratulate from bottom of my heart to Sachin Salunke, sir. Okay. So it gives me an immense pleasure to share some of my views with you all on this closing occasion of our 4-day e-faculty development program. So let me thanks first our management, the entire team for their support, their guidance to organize this mega event. And during these 3 days, you have sincerely attended the scientific deliberations on the theme Pharma Industry Insights, Recent Trends, Innovations, and Digitalizations. So each of our speakers, they have shared their experience, their knowledge, and presented their views, which have, I think, more than 100%, they have matched with our theme for this 4 days faculty development program. So many of the participants, they have joined from different parts of our country. I also congratulate them because they are taken up such information which is vital for them and to address their own students at their college levels. So I am very much thank you to all the participants on this occasion. And all the participants, since from the first session to the last session of today in the scientific deliberations, they have raised some queries, and our speakers, they have addressed all those particular queries. So this indicates me that all of you are much more alert, and your active participation is going to observe through this particular your participation. So this particular situation is arises because of COVID-19 pandemic, no doubt. Every aspects, every departments, they are facing the problems and issues. So we, in the education department also, we are also having this particular effect of pandemic COVID-19. But the lesson from this particular organization, we can learn here that now we have to shift our gears from the actual classroom teaching, that is a chalk and board teaching, to we have to divert to the online learning and teaching concepts. And for that, we require ourselves to develop in that particular fashion, because this particular pandemic, COVID-19 pandemic, it has changed our lifestyles also. So in this case, we have to move forward. We have to learn so many techniques, and it may be possibly difficult for the persons like us having at the verge of retirement. But I am seeing the participants, they are quite young, they are dynamic. So it is always a responsibility of you people to take it as a challenge and try to develop our students, as Sachin sir has shown his last slide, that this particular digitalization is going to affect our students' career also in future. So definitely we have to mold our students. We have to make them understand all these particular concepts which will be helpful for them in a digitalization process is going to happen in our future. Similarly, I know you all are capable for that and you will do that particular noble job of imparting such things to your students and motivate them. So this COVID-19, no doubt pandemic situation has developed so many crises. There are so many cons, but at the same time, it has given up a lot many opportunities. And these opportunities we have to grab. And to grab these opportunities, as I told you, we have to uplift ourselves. So don't hesitate to take the information, the knowledge, whatever the source may be. Just you have to assure that the source of knowledge you are getting, whether it is a genuine one or not, because there are so many fake news which are circulated amongst the WhatsApp and other social medias. So definitely we have to take care of that. And at the same time, we must learn all these platforms available for LMS, that is a learning management systems. So once you acquainted with this particular LMS, so definitely we can guide our students, we can motivate our students, and we can make them competent to fit into the dynamic nature of our healthcare system, where they are going in all version of digitalization. So definitely, I believe in you, your strength, and we can produce good citizens at the same time, more importantly, a competent professional pharmacist in future. So one particular things which I generally don't want to express myself, but the all these particular sessions which I have observed, so just I want to leave a message here. Those participants, no doubt there are very active participants, they are always alert, but at the same time, there are so many participants which I observed, they are joining at the end moment, possibly uh, it is my imagination, I don't know exactly, but don't attend such type of informative scientific deliberation for getting the certificates. So it really hurt that participants, they are coming in, they are going out and again joining. So it shows that there is a certain causes behind these particular things. So ultimately, suggest the participants, whenever you attend such type of online versions of scientific deliberations, try to understand, try to be active in that, and try to listen what speakers are saying. So definitely, I am proud that the Sant Gajanan Maharaj College of Pharmacy has organized this faculty development program on online mode, and there are a lot many hands behind this to make this particular program a successful one. Our core team, our committees, our management, they are all fully supported and they have worked day and night to make this particular program a grand success. So I thank you, all of them, and I thanks all the participants from all over the country. And possibly I want to stop here and we can proceed further. Over to Samir sir.
Thank you, sir, for sharing your valuable thoughts and addressing the participants. Now I request Professor Alman sir to propose a vote of thanks. Over to you, sir.
Good evening, all of you. Respected trustee, directors, principal, delegates, teaching and non-teaching, and participants from different colleges and universities. Sant Gajanan Maharaj College of Pharmacy, Mahagaon, organized 4 days national level e-FDP program 2020 on pharma industry insights, recent trends, innovations, and digitalization. Myself, Mr. Abhinandan Alman, Department of Pharma Chemistry, feel honor and privilege to get the opportunity to propose a vote of thanks on this special day. First of all, my great thanks to Advocate Dr. Annasaheb Chavan sir, Chairman of Sant Gajanan Maharaj College, Sant Gajanan Maharaj Education Group, Yashwant Chavan sir, Sanjay Chavan sir, and other trustee members of SGM Education Group to give us a platform and be with us as a motivator. Thank you, sir, for the moral support. I must mention our deep sense of appreciation to expert speaker, Mr. Chetan Jadhav sir, Mr. Subhash Patil sir, Dr. Basuraj Nanjwade sir, Dr. Santosh Vankudre sir, Dr. Santosh Gezge sir, and Dr. Sachin Salunke sir. Thank you very much, sir, for accepting our invitation as an expert speaker for this national level e-FDP and spent your valuable time with us by sharing your great knowledge and views through online mode. My special thanks to my very dynamic and versatile personality of our principal, Dr. S.G. Killedar sir, for excellent guidance throughout this FDP. I would like to thank the organizing team, conveyor, co-conveyor, coordinator, and all staff members for the smooth conduction of this e-FDP. My sincere thanks towards all my faculty members who have been played a great role to various committees and be a part of this e-FDP directly and indirectly, but having a contribution in making this event a grand success. Last but not the least, I'm very thankful to all delegates and participants who were present and attended this e-FDP directly through online Zoom meeting or else directly through indirectly through the YouTube streaming for this entire knowledgeable session. We hope that you have been taken a lot of knowledge or upgrading your skills throughout this e-FDP. So once again, I'm thankful to all of you. Thank you. Thank you very much.
Thank you, sir.
Over to you, Nadav, sir.
Yes, yes, sir. Okay, thank you, sir. Now I request all the participants to turn on their video mode. So we'll have a photo session with our esteemed speaker for our memory as well. Request Gadge sir to proceed with capturing the photos. It is Gadge sir, it is over.
Done with the photo session. Nadav sir.
बोला सर.
Okay, photograph has been captured.
Okay. Hello, sir.
Yes, sir.
I think photo has been can proceed with.
Yes, sir. You are not audible, sir.
Photo has been captured. You can proceed with the.
Yes, sir. Yes. So thank you all the participants. Now I would like to give you some instructions related to the exam. As you know, we are conducting the non-graded examinations. In last 2 days, you have completed the 2 non-graded assignments. So today as well, you are going to have a non-graded assignment, and you will get that within 1 or 2 hours. And as you know, tomorrow there is a graded assignment. So that schedule will be discussed in WhatsApp group, as well as the graded assignment will be shared tomorrow from 10 a.m. onwards, and details will be shared in WhatsApp group. So do not miss that graded assignment. The graded assignments will be shared, and you need to score minimum 50% mark. So your attendance of this all the sessions, as well as the marks in graded assignments, will be considered, and then only certificates will be issued. And do not bother about the attendance of today's session. We have collected your attendance is with us, so do not bother about that. So with this note, I conclude this session, and thank you for joining with us and making this event a mega event. Thank you so much. Now I conclude to all the speakers as well as participants for joining with us and making this event a mega event. Thank you so much. Now I conclude this meeting. Thank you.
Samir sir. Hello.
Samir sir. I think sir has left.
Okay, okay. We'll leave it and I will call you. Okay.
Okay, no issue, sir. Okay, thank you all of you. Thank you for joining.